So this is a continuation of a presentation that I talked a little bit about back in December. So I'm going to cover kind of the second half of it this time, and that's going to be more about the actual networks themselves. But of course, when I talk about networks here, um, in a way, am I actually getting things started here? Um, anyway, as I mentioned before, networks are kind of an easy way to say this is complex. We like to think about these graph structures where everything's integrated with everything else, and that'll allow us to explain some additional level of complexity in data that otherwise isn't connected. And while a lot of times this is kind of a shorthand, at least once we get this to the stage of being a finished product that we're actually showing off to people, uh, in many ways that's actually true. It's these relations between individual elements, whether it's in biology or beyond, that really makes them understandable to us, but also reveals a lot of the underlying phenomena that we can actually generalize about in useful ways. And it turns out in biology in particular, this is an area where we have a lot of experience. Even if we're thinking about something like a metabolic network, we realize that this isn't just a bunch of individual metabolites and enzymes just sitting there and acting at random. They have very regimented and organized ways in which we can at least understand them to operate. In practice, they don't always operate in the organized fashion that we'd prefer them to, at least in a way that would enable us to understand them better. But we can this enables us to form these metabolic networks and understand the relations between things. So networks aren't really a new concept in understanding biological phenomena, but they're an area where we are continually gaining new ways of understanding them and about generalizing them in useful ways. Now, of course, there's specific challenges that we run into even when we're just looking at networks in a particular field. Like, let's say we're looking at cardiovascular disease research. If we want to be fully comprehensive about, let's say, just looking at a network of protein-protein interactions, we'd say, okay, well, if we're looking at human disease, we'd want to look at every last protein in the human system or every last protein in the human heart and ask, okay, how are all of these proteins interacting? And in practice, that's probably overkill because we're already talking about the proteins from more than 25,000 human genes. Some of them are going to have different properties that we may be more or less interested in. We're going to have different isoforms of those particular proteins that may interact in different ways or with different partners. Some of those are going to be from essential genes, so potentially they're more important. So right away, we begin to run into issues of complexity, even within a small network that we need to deal with that, on one hand, can reveal some really interesting phenomena, but on the other hand, become quite computationally and, of course, biologically complex to have to deal with. So even just comprehending this in kind of the level of the underlying network becomes quite challenging. So one way that I'd like to think about this, and I introduced this in the last talk, was the meta-interactome. In short, this is just the idea where if you have a set of all the protein-protein interactions or your interactome in a given area, then in many cases you want to get the interactions from multiple studies, from multiple organisms, from multiple sets, and essentially just compress them into a smaller network where you have something similar and you have kind of these larger, more compressed nodes. I tend to favor orthologous groups because then you can observe data from a number of different model organisms. But essentially what you're performing is dimensionality reduction in the end. You just have less to worry about in the long run. Now, of course, this does mean that you can lose a little bit of resolution in your data at some point, and that may mean that you're missing some potentially important biological phenomena, but it makes it a lot easier to handle that underlying network and potentially even retains a lot of the underlying phenomena that are more interesting to look at in a cross-species perspective, because that's often what we're really more interested in. We don't want to necessarily lose all like the great mouse model data that we have, and we certainly don't want to do all of our new physiological observations in the human model, because that's just not feasible. So last time I also mentioned that I have a script that I've been working on to deal with some of these problems, and this is a work in progress, but if you are interested, then this is on GitHub as the Pining project, or Protein Interaction Networks and Integration with Novel Graphs. So right now, this mostly just works to do some of the compression work and retrieving interactions from databases and such. But working on looking at those subgraphs or the smaller components of the resulting protein-protein interaction graphs to try to see if there's something something interesting about those subsets of interactions. And luckily, there's been a lot of very interesting work in this field, so that I can borrow from. But if we're looking once again just in the span of, say, cardiovascular disease, and I apologize for some of the odd fonts here. But if, let's say, we wanted to assemble a cardiac interactome, I said that it's really challenging to assemble all the protein-protein interactions for one particular species, even if it's humans. But in practice, if we can get many interactions for many proteins, especially if we already know the proteins that we're interested in. Say here, we have a recently produced data set out of our lab dealing with cardiac proteome turnover, or essentially the levels of proteins in the heart that we may have at a certain period in time, and how that's changing over time. So that gives us a set of, in this case, more than 8,000 proteins to work with. We can compress those further on the basis of orthology. We can compare those in a network. We can derive from that protein-protein interaction data from multiple data sets and assemble those into an interaction network. And that actually ends up being quite large, so we have to filter it a bit further. So once again, this is something that I mentioned last time, but we can even just focus on some of the proteins that have multiple isoforms but seem to be predominantly interacting with a number of different cardiac proteins. And in this case, once we match that up with the cardiac proteome turnover data, we begin to see some interesting phenomena related to 14-3-3 proteins. So these are hubs that not only seem to interact with a number of different proteins, but some of the proteins that they interact with have noticeable differences in turnover when you compare them between control and a model of cardiac hypertrophy. So these are potentially proteins that are directly related to some potentially pathogenic physiological phenotypes, which can be quite interesting, especially if we follow them up, because that's narrowing down our set of more than 8,000 proteins to just a few that we can focus on. But what I'd really like to talk about a bit more today is how these networks are useful in looking at literature. So I went into some detail last time and this time about protein interaction networks, and about how these are essentially just networks where we start with a list of proteins, potentially whole proteomes with the proteins. We combine them with their features and their relations between those proteins, in this case, often protein-protein interactions. So that leaves us with a graph structure of nodes that are proteins, edges that are interactions between those proteins, and we can compress them into orthologous groups. Now, if we do that, I call the final product there a meta-interactome. But we can do something very similar with text documents as well. This is especially useful for technical documents, like, let's say, medical case reports, which I'll get into in a bit more detail shortly. But then if we take the entities or the distinct concepts in each of those documents, those become the nodes in our graph. The edges can become dependency or proximity in text. There's some relation between those entities and concepts. And then we can compress that further in much the same way that we'd produce a meta-interactome into a higher level representation of each entity or concept. We kind of unify it across those documents, and what we end up with is a knowledge graph. So what is a knowledge graph in practice? Because I certainly didn't invent that concept. In fact, this is something that people have been working on for a number of years now. So here's kind of an example of the general idea here, where you have a graph of those entities. Well, you're unifying them on the basis of these relational facts. These are usually things that can be expressed semantically. Traditionally, we think about something like a knowledge base, where we essentially just have a collection of information about a number of different topics, potentially otherwise unrelated topics. But in a knowledge graph, we can take these entities, like, let's say we have Bob Dylan here, and we know a number of different facts about him, but these are facts that are also shared with other concepts, like the fact that he's certainly not the only person who ever lived in the United States. And even beyond that, there's a number of different places in the United States. They're all located in the United States. So that's the relational fact there. And then, of course, each of those states has other cities, in turn, that are located within them. So by taking these relational facts, we can establish this this knowledge graph, or this collection of otherwise heterogeneous information, and unify these concepts in a way that gets complex quite quickly, but is also very useful for unifying otherwise disparate facts that especially would be much more easy for a computational assessment. Now, of course, search engines and the companies that run them in particular are very interested in this concept. And in fact, Google has been using what they just call their knowledge graph since about 2012. And in fact, if you go on Google right now and you search for heart attack, then you will see this card shown on the left. And that's directly the result of their knowledge graph kind of idea, in that they don't recognize that this is just an important phrase or a known entity, but that it is a phrase that has other not only related properties that they can look up in their knowledge graph, such as the fact that it's a very common problem, it requires a medical diagnosis, it usually occurs when a blood clot blocks blood flow in the heart. So these are things that you need to be able to look up if you're doing this computationally. But perhaps more interestingly is what's shown on the right, and that's if you scroll down to the bottom of that card, you see related conditions. So these are all neighbors in the knowledge graph, or in this case, conditions that are similar potentially in a number of different ways, not just in terms of sharing the same symptoms, but potentially in the fact that they're common. They may affect a lot of the same populations. So because of that knowledge graph, we can unify things in a way that really shows us what kind of entities are really similar and how. Now, of course, we can look at these knowledge graphs in a very general sense and say, okay, well, there are connections between just about as many different entity types as we can find, but we can also make them more individualized and say, okay, we're going to look at this in terms of, say, individual patients, where each patient has a set of entities that apply to them. Perhaps we're looking at a electronic health record where we know that a patient had a certain set of symptoms, they were taking a certain set of drugs. We can draw connections between these things, but then we compare those not only to other patients, but to potentially also to published reports or other sources of data, even other biomolecular sources of data. If we have experimental data, that also presents its own potential source of an individualized knowledge graph. So these aren't necessarily concepts that have to be taken all at once. We can take certain subsets of them and compare them and really find out what's particular about them that makes them unique. So I mentioned case reports a few times, and I like to think that these published case reports are a really good way of understanding not only the individual documents that might go into knowledge graph if we're especially interested in looking at medical or clinical data, but they're also kind of models of this just kind of concept of a medical narrative. So when I'm specifically talking about clinical case reports, I'm talking about the published case reports of individual medical situations or cases, generally available through PubMed. So these are generally publicly available, notwithstanding any copyright issues or paywalls or such, but they're not electronic health records, so they're not necessarily subject to all the privacy concerns and HIPAA regulations that go along with that. But I like to think that we can look at an entire clinical case report corpus in terms of its individual documents. And then for each one of those individual documents, we can break that up into 3 different types of document metadata. Some of these are document features, where we're looking at things like the title or features of the patient involved, their demography, maybe the general idea that we have about their case. And much of this is already provided through PubMed through mechanisms like the MeSH terms or the medical subject headings. So that's all fairly high level. If we drill down a little bit farther beyond that, then we can get more detail about those concepts. So at this point, we're talking about concept level features. And we can learn things like, okay, we weren't just talking about a woman in a particular age group, but specifically a 75-year-old woman. And we can get more details about those signs and symptoms and such as well. Such in this case, they presented with shortness of breath, they had chest pain. So at this point, we not only gain more detail, but if we ask something like, okay, what were the signs and symptoms? What were the drugs involved in this case presentation? What were the eventual diagnoses that the clinicians prepared? Then we can get short answers that define those concepts. Now, of course, we can go even farther beyond that and look at that at the text level and identify individual events and entities within those concepts and say, for example, well, an X-ray was done, perhaps a chest X-ray. It showed a particular thing. The clinicians performed a particular therapeutic procedure, and then after a certain amount of time, they made a clinical assessment. And they realized in this case, okay, the patient was doing well at the point of a 6-month checkup. So at this point, we're not even just identifying exact entities that are present, but actually their relation to those higher level medical concepts. Now, I'd like to think that looking at case reports in particular is and will continue to be especially useful because in PubMed, at least as of last fall, in total, there's well over 1.8 million of these documents. There's tens of thousands of them published even on a yearly basis. We thought for a while they seemed to be declining in trend, and that's kind of what this graph would imply over time. And it's possible that it's more that they're just kind of leveling off because sometimes there's a year or two delay in actually getting all these case reports on PubMed and getting them indexed and properly available to the reading public. But in practice, this is a massive source of these model medical documents that we'd like to think is not only a good source of training data, but also a good source of interesting medical observations in its own right. So I will refer to this kind of general assessment of the concepts in clinical case reports as annotated clinical case reports, or ACCRs. So right away, even just looking at these documents, we can start to answer those document level questions related to the document level data types. These could be questions like, what are the most common subjects in a particular group of documents? Or where do these particular clinical narratives take place geographically? If we're given a set, those 1.8 million of them, how many occurred in North America? And then we can correlate those with other details like, okay, out of those that occurred in North America, how many of those were related to rare diseases? How many of those were male versus female? What kind of age groups are we talking about here? So these are those highest level concepts, but they're still very interesting once we start looking at this big data collection. Once we break that down to the concept level questions, we can start to ask things like, what kinds of subjects are included in a medical history versus maybe what should be included? So we can start to ask things like, what's missing from here that we might expect to see? Especially if we're comparing this across a number of different documents, we might realize, say, 9 out of 10 documents will provide a clinical assessment at the end of their particular narrative, but maybe that one document doesn't actually reveal what happened to the patient. And we can follow up with those individual documents and realize, okay, perhaps that's because the patient didn't want to return for treatment. Maybe they actually didn't have that good of a clinical diagnosis in the long run, or maybe the particular publication where these case reports are being published just doesn't mandate it. Maybe it's of lesser interest in the long run. Maybe they're really focusing on the novel observations in that report. So there can be a number of different reasons that we can look into there. Now at this point, this is especially useful for things like distantly supervised learning, because at this point we can think of it as a computational task. So computers, we know, can do named entity recognition pretty well. That is, we can tell them, okay, we have this list of concepts like heart attack that we want you to be able to find in a document, but they don't always do so well with the context around that, like what happened immediately before or after a heart attack, both in a semantic sense, in that actual document text, or even in a temporal sense, for the actual events that occurred in the course of that clinical narrative. So at this point, I'll talk a bit about how biomedical documents are actually parsed in practice. I've alluded quite a bit to PubMed as being the primary source for these documents, and since they are such a large source of biomedical documents, they have some extensive experience in parsing them and preparing them for entry into their database and making them nicely indexed and searchable. And of course, I really appreciate all these efforts. It's really quite amazing what they've been able to accomplish. Um, but of course, this is a very complex workflow that they have. What I've shown here is what they consider their the National Library of Medicine's medical text indexer that does a lot of the heavy lifting. This certainly isn't specific to clinical case reports, but rather ends up working on much of the biomedical document that enters the PubMed. But it's especially reliant upon these medical subject headings. And that's because much of what they're trying to do is indexing. They're essentially trying to ask, what are the critical concepts in a particular document? What is it really focused on? What are the major ideas that are covered in it? So if I'm looking at a particular biomedical document and it's mostly about heart attack, but also mentions a particular drug at some point, it may miss that particular drug is coming up because it's not one of the primary subject headings. Now you also notice that right at the top of the stack, there's a couple steps that are working with a PhraseX and MetaMap. So some of the initial steps in looking at the title and abstract of every document that goes into PubMed is that they need to search for particular phrases. So that's part of this named entity recognition portion. And based on those phrases, they can look up those higher level concepts. But beyond that, they're not going through every last individual medical concept or medical term in any particular biomedical document, because that would be excessive. Unfortunately, that's the kind of thing that we are interested in, because we'd like to think that we can do a lot of really interesting things with those case report text features. So that's specifically, again, that what I've highlighted in red here, or these kind of the lowest level concepts that are still most interesting in context, but also capable of showing us a lot about the actual progression of events that occurred within a particular clinical narrative. So back to how clinical text is usually parsed in practice. There's a long history of mechanisms that people have used to specifically look at more technical language in particular. Some of the first steps usually include pre-processing it. You need to do things like controlling for punctuation. Technical language usually has a lot of abbreviations that you may need to handle or at least define, or those known constructs such as known named entities, particular keywords that have some kind of higher level meaning. So this is all often done at a pre-processing stage. And then once you actually do the bulk of the named entity recognition and semantic tagging, then you may go through and find things like, okay, acute coronary syndrome is a recognized phrase. We know what this means, and we can link this through documents. The NLM often does this using their own UMLS vocabularies, and they link these through what they call MetaMap. But this can also be done through other established ontologies, such as mesh terms, as well as medical coding mechanisms like ICD-10, ICD-9, as long as you have some kind of dictionary of terms. That's essentially the minimum that you need to look up those terms in text. The next step is often to do some semantic typing, which may mean that you need to actually identify what type of thing in a particular ontology each of those entities is. So in this case, we may recognize that acute coronary syndrome is a disease or syndrome, because that's what the UMLS refers to it as. In MESH, we know that there's more of a hierarchical definition, so we recognize that it's a subtype of heart diseases. Computationally, both of these are very interesting things to know, and at the very least, they give us a way to organize all of those named entities that we recognize in the text. Beyond that, we may want to do relation and event extraction, and in fact, in practice, this is also a very interesting thing to be able to extract from a clinical or medical document in particular, because we can say things like, okay, a 42-year-old male, so we know we have a male patient, presented with acute coronary syndrome. So a patient presented with, that's the relation, a disease. And we can go through an entire document and extract things like that, depending upon the types of relations that we're looking for between our entities or events. But kind of the critical aspect throughout all of these methods and beyond is human oversight. Right now, all of these kind of efforts need extensive oversight by experts in many cases, or in some cases it's done as more of a crowdsourcing problem, but we always need some kind of human oversight of the results that we get out of these systems. They're simply not good enough for our demands. Sometimes it's a matter of mistyping a particular entity recognized from the text. We may look at something and think, okay, this is a drug, when in reality it's actually a disease, or perhaps it's a particular compound that's not actually being used as a drug. So there's a lot of idiosyncrasies that we can run into. And sometimes it's really just a question of whether the annotations that our computational systems are making just even make sense. Now, there's been efforts such as the I2B2 challenges that have been really helpful in providing a bunch of the training data that people have used to develop some of these methods, as well as some larger platforms that I mentioned here, such as C-Takes and M-Terms. More recently, there's a smaller platform called Canary. But in practice, there really aren't that many platforms out there that will take a complete collection of medical documents or even clinical case reports and extract quite the level of detail that we'd like to see from them. And certainly none of them that I know of that assemble them into a knowledge graph. So in practice, we consider this to be an information extraction problem. We're not really just interested in knowing what a particular report is about, but really we just want to impose some kind of structure on its concepts. We're trying to take unstructured clinical text, impose a structure on it beyond its major subjects. We want to know more or less its complete contents. So if we're identifying features of the events in a particular clinical case and the relationships between those events, as long as we also have the specific features of those events and of the patient, then we can start to assemble this knowledge graph in both kind of that individualized way on the set of a document or a set of documents, but also for our entire corpus of clinical case reports. This lends itself pretty well to graph representation. As I mentioned earlier, that's essentially just when we take those entities and we link them on the basis of those relations. And what we end up with is this heterogeneous information network. Now, in practice, what our annotation may look like and what our interface does look like is what we show at the bottom here, where we actually highlight the particular concepts in text. So that's our text level annotations. The concept level annotations are broader than that. But in practice, at the moment, we are, in fact, preparing a corpus of about 200 documents, and we're actually getting pretty close to finishing that with text-level feature annotation. We're using the BRAT annotation interface for that, so that's open source and very convenient to use. But we're working with Wei Wang and Yizhou Sun's groups here at UCLA to develop new methods and platforms for doing that, both in a more organized way in terms of an ontology and developing some computational methods that will make it easier to do much larger sets of documents. And we're also working with Xiaowei Han's group at UIUC to do some more in-depth language processing and data analysis, along with some very interesting distantly supervised methods that they made some great innovations on, even just in the last few years. We also have an ontology of about 40 different labels and 12 different relation types that we're using to keep these clinical cases and their individual text components more organized in a way that makes sense to us medically. But in the long run, what we're really interested in doing with all this data, once we get it past the standardization step, is to generate this annotated clinical case report corpus and then use that for 3 primary goals. One of which is to just get a general model of medical language. So this is not just the individual components in any particular document or set of documents, but really how medical language is used in a clinical report context. And that could be extended to understanding things like electronic health records much better. We want to integrate this with biomolecular data, as I mentioned earlier about the individualized knowledge graphs. There's certainly a number of different sources of data that we'd like to be able to integrate in a way that will enable us to better understand things in the context of actual biological phenomena. Lastly, we like to think that Case reports in particular provide what we think of as a virtual cohort. This could be particularly interesting in looking at rare diseases, because in a number of cases, the clinical case reports may be the only place where these rare diseases have been described. If we're talking about something where cases only occur once in every 300,000 people, then they're rare enough that even if you've been doing medicine for decades, you may never see a case of this. So clinical case reports may be the only place where we have a description of the case progression, and hence something that we can work with if we're studying rare diseases in particular. So I mentioned that there are some other platforms that do some similar things to this, and we've been borrowing some concepts from the CTAKES project in particular. More recently, there's actually been a platform released called CLAMP that does some similar things, but not quite the same as what we're doing in terms of knowledge graphs. But in more of a machine learning concept, and this isn't really a platform so much, just something that came out last year and was interesting. Andrew Ng and a number of colleagues in the Stanford machine learning group took a large set of medical records and extracted some predictions on how likely patients were to die within a given period of time, given knowledge of particular clinical features that they had. And they got some very interesting results, but this is where much of the field is going at this point, is using machine learning, and more particularly deep learning methods, to understand much of the clinical phenomena that may be presented in the form of a case report or an electronic health record. Now, what I think that project in particular really illustrates is that much of the effort that goes into these larger projects is still about processing the data and determining which features are really going to be useful, and about removing data that you don't think is going to be contributing anything of use at all. So there really is a lot of human pre-processing, human oversight, human annotation that goes into these projects. It's not like we can just throw deep learning at it and say, okay, we're done. Tell me what's interesting. At least not yet. That may be different from the opinion of some of the larger companies and health service providers that have also been working in this field of applying machine learning and data analysis to natural language processing in the medical domain. Optum Labs is one group where they've been working with a product that they call Optum Life Code. I think that is particularly focused on medical coding. There's a UK-based company called Linguamatics that has a product called I2E that does medical natural language processing. They claim that that's scalable to every document in Medline. I don't know if that's actually true or not, but they apparently made some progress. And also the Geisinger Health System has their own NLP platform where they're doing language processing with free text electronic health records. So they've also made a lot of great strides in this area. So it's a very interesting field to work in, and I hope that some of these more proprietary efforts will release their findings, perhaps in a way that will benefit health and at least help us to better understand the underlying biological phenomena. So what's really unique about this approach, at least what I think will be interesting, and what I think will hopefully lead to some useful next steps, are that we're taking a graph-based approach to understanding clinical narratives. We're developing a clinical case report corpus of annotated reports that include varied medical observations that aren't restricted to any particular field or area or privacy restricted, unlike electronic health records most of the time. And we'll be developing some distantly supervised machine learning approaches to doing this medical text processing. This will enable us to do some integration with diverse biomolecular data sets and eventually develop a broadly usable platform applicable to both clinicians and patients to better understand, in particular, rare diseases, but essentially any set of biological phenomena that may be expressed in a clinical case report. So with that, I'd like to thank everyone on this slide, in particular the Ping Lab here at UCLA, as well as our collaborators at UIUC and at UCLA as well, including everyone else in the Heart BD2K Center and the BD2K CCC. With that, I will take any questions.
Thank you, Harry. Do we have any questions?
Harry, this is Andrew. I just have one question. First, as a great talk, this really clicked for me this time in terms of the clinical case reports as a corpus that could be really valuable to mine and query. So as a resource to provide to the community, can you talk about the plans there, to essentially expose the knowledge base to others to query?
Right. Yeah. I'd like to make as much of this open as possible, and I I think that the actual graph itself, in practice, may not be that useful to people. So I think a lot of the effort that we're going to have to put into this is in providing an analysis interface for people, because I think this is the kind of resource that will be most useful to people in the way that they are used to understanding it. Because if we give people a set of data that's based on 100,000 documents, I don't want them to have to go back to each of those 100,000 documents just to understand the context of things. So I think this is going to be a challenge. Is kind of the short answer here.
Sure. I would push back a little bit to say that yes, to meet build a query interface that is accessible to clinicians and non-computational savvy researchers. Yes, it makes sense to build an interface on top of that, but exposing the underlying knowledge base itself is also valuable, both to computational scientists who want to mine that data, and also to other data, other people who want to build interfaces that may be complementary. I mean, I think that there's the number of applications that could sit on such a knowledge base. There are many different takes on that. So
certainly,
I would encourage you to, put it this way: if the underlying knowledge base were exposed and released and either downloadable or accessible via an API or a graph database, please let me know, because I think that'd be really interesting to look at.
Right. Okay. Yeah. No, I'm glad you bring up the just exposing it to the APIs, because I think that's possibly the easiest way if we're integrating it with especially something like the BioThings API, where it's like, okay, we can, if we have a kind of a universal API that we can already play with, then that certainly saves a lot of work for us. And yeah, I'm certainly glad to share as much of it as possible. But yeah, I'm just trying to stay very aware of our audience as well. But yeah, thank you.